The overall goal of this procedure is to show how to dissect and prepare a mammary gland whole mount and analyze its morphology to predict mammary cancer risk. This is accomplished by first dissecting a rat abdominal mammary gland and then preparing a mammary gland whole mount from the dissected gland. The next step is to analyze the mammary gland morphology made visible in the whole mounts. The final step of the procedure is to palpate a rat for detection of mammary tumor and to measure the tumor volume in millimeters using a caliper. Ultimately, results can be obtained that show changes in mammary gland morphology through identifying the structures in the mammary whole mounts and counting or scoring their amounts under a microscope.
This method can help answer key questions in early life exposures and breast cancer field. Questions such as how dietary or environmental exposures acting during development can affect mammary gland differentiation and thus breast cancer risk later in life.
The implication of this technique extend also toward breast cancer prevention. We can study, for instance, environmental and dietary exposures during the development and see the changes in the mammary gland, whether they are related to increased or decreased breast cancer risk.
Demonstrating the tumor palpation procedure will be Idalia Cruz, an animal technician and manager for Animal Shared Resources.
To begin the procedure for removing the mammary gland, first use needles to pin the euthanized animal onto the surface board on its back from its legs. Then wipe the skin wet with ethanol. Lift the skin a little with forceps and with sharp scissors make a midline incision into the skin starting between the pair number 5 nipples and cut toward the neck. Then make an inverted Y incision from the midline toward the hind legs. Dissect the skin open with scissors on one side of the inverted Y incision to expose the abdominal number 4 mammary gland, which is attached to the skin. Pin the skin onto the surface board with needles to completely expose the gland. Next, use sharp scissors to dissect the mammary gland free from the skin. Start from the proximal area close to the nipple and work toward the distal end of the gland toward the spine of the animal. Once the mammary gland is detached, immediately spread it onto a glass slide for preparation of a whole mount. The glass slide must be bigger than the gland and appropriately labeled using a permanent marker and/or a diamond pen. Spread the gland carefully corresponding to its original size and shape in situ. After spreading the gland onto the slide, let it sit on the table for about 30 minutes so that the gland sticks onto the slide, but do not let it dry. Next, put the slide into a jar containing carnoise fixative and leave it at room temperature in the fume hood for 2 days or longer. After at least 2 days, wash the slide in 70% ethanol for 1 hour at room temperature. Rinse the slide in distilled water for 30 minutes at room temperature. Next, stain the gland in carmine alum stain for 2 days or longer. Staining is done when you check the back of the slide and see that the lymph nodes have stained through. Wash the slide in a series of increasing ethanol concentrations each for 1 hour. Start with 70% ethanol, followed by 95% ethanol, and thirdly in 100% ethanol.
To obtain mammary gland whole mounts that are technically good for morphological analysis, 2 steps are essential: dissection of the whole gland and the lipidation of the mammary fat bed. The gland is placed in xylene to remove the lipids and to increase transparency. This clearing will take place 2 days or longer, depending on the size of the gland and how much fat there is.
After the last wash in absolute ethanol, put the slide in xylene. Allow the gland to remain in xylene at room temperature for at least 2 days. Once delipidation is complete, mount the slide with a cover slip using mounting media such as Permount. Let the slide dry for several days in the fume hood before observing it under a stereo microscope. The morphology of the mammary gland whole mount is analyzed according to 3 endpoints: epithelial elongation, number of terminal end buds, and differentiation. The results are then correlated with mammary cancer risk. We will first demonstrate the analysis of a prepubertal age mammary gland whole mount. To access epithelial elongation, use a ruler to measure in millimeters the distance from the nipple to the end of the epithelial tree. The second endpoint, number of terminal end buds, or TEBs, reflects the potential for malignant transformation. Under a light microscope, count the number of TEBs, which are the largest bulbous structures located only at the distal end of the mammary epithelial tree. For the third endpoint, which is differentiation, the whole mount is also examined under the light microscope. Differentiation is measured by scoring the alveolar buds, which are spread across the epithelium. The two alveolar bud types, designated AB1 and AB2, are each given a score from 0 to 5, based on their abundance in the gland. The score values for AB1 and AB2 are then added for a final differentiation score. The morphological analysis of a postpubertal age mammary gland whole mount is similar to that of a prepubertal age mammary gland whole mount with the following differences. To examine epithelial elongation, first use a ruler to measure the distance in millimeters from the lymph node to the end of the epithelial tree. Second, measure the distance from the tip of the epithelial tree to the end of the fat pad, also in millimeters. For analysis of differentiation in a postpubertal age mammary gland whole mount, in addition to alveolar buds, lobules are scored on a scale of 0 to 5 under a light microscope. The score reflects the abundance of lobules in the gland. Differentiation is then assessed in two ways. First, the score values from AB1, AB2, and lobules are added for a final differentiation score. Second, the ratio of the lobule score to the AB1 plus AB2 score is calculated. Since ABs differentiate two lobules, a more differentiated gland will have a higher ratio of lobules to ABs. To begin this procedure, hold the rat by grasping the whole body with the palm over the back, with forefinger behind the head, and the thumb and second finger under the opposite axilla. Turn the rat so it is lying on its posterior, and palpate to detect any mammary tumors. Palpable tumors should feel like a lump. Next, use a caliper to measure the width, length, and height of the tumor. Calculate the tumor volume using the ellipsoid formula. Volume equals 1/6 pi ABC, where A equals width, B equals length, and C equals height. Manually record the location and size of the tumor in a notebook on a weekly basis. This data will be transferred to a spreadsheet later. When dissected and processed correctly, the mammary gland whole mount should have all its components clearly visible, as shown in this example. In contrast, this is an example of a poorly prepared mammary gland whole mount. The gland has not been properly dissected because the distal portion is missing. In addition, the gland was not properly stretched and delipidation has not occurred completely. Shown here are representative results of how changes in mammary gland morphology can predict mammary cancer risk in rats that were exposed to the carcinogen DMBA. When the number of terminal end buds, or TEBs, was counted, the treatment group had a higher number of TEBs at 21 days compared to the control group. These changes in mammary gland morphology were associated with a higher proportion of mammary tumors in rats of the treatment group, compared to the control group, as shown in this graph.
While attempting this procedure, it is important to remember to dissect the whole mammary gland and to remove all lipids to ensure good visualization of the mammary gland structures.
After watching this video, you should have a good understanding how to dissect and prepare a mammary whole mount and how to analyze its morphology to predict breast cancer risk.